That's great. Yeah. Hi. So previously, under Henry's leadership, the CFC developed the Viper for RNA-seq analysis. As you can see, recent years, the cost of RNA-seq has decreased continuously. Now we can do RNA-seq at CRO company for like $160. And yet the information within RNA-seq is so rich and versatile. And so we have, within the lab, developed a new pipeline called RIMA, which is, in addition to the standard differential expression pathway analysis, a lot of other features with tumor immunity. And we hope this would be very, very useful for the community and CFC who are interested in tumor immunity. So without further ado, I'll give it to Yang.
Yeah, thank you, Shirley. Let me share my screen first. Yeah, am my screen viewable to everyone? Okay, then I'll start. Good afternoon. My name is Yang Liu, and I'm a scientist at Dr. Shirley Liu's lab. I joined Dana-Farber in early 2020 after graduating with my PhD in bioinformatics from University of Missouri. I had experience that analyzing multiple NGS data, and I also have a background with machine learning and deep learning to discover the interesting biomarkers that is associated with the important phenotypes. So I had tried to use the convolutional neural network on the SNP data that identify the markers that are associated with the I used to work on soybean, that is the yield traits. So now I'm actually leading the RNA-seq pipeline development in the CIDC that I'm presenting today. Before we go to the pipeline, I may introduce a little bit about what is the CIDC network. It's actually called the CIDC network, and the CMAC stands for the Cancer Immune Monitoring and Analysis Center. CIDC is the Cancer Immune Data Commons of this network. So this CMAC CIDC project is sponsored by NCI as well as the industrial companies as part of the PACT funding for the partnership of accelerating the cancer therapy. So under the CMAC CIDC network, there were 4 centers, with mainly the Dana-Farber, Mount Sinai, Stanford, and the MD Anderson. We are the bioinformatics center that is responsible for the bioinformatics analysis using the data delivered by those 4 CMAC centers. Within CIDC, we have several pipelines that are crossing the whole exome analysis, ChIP-seq, and the TAC-seq analysis, as well as the TCR-seq. Today, I'm going to only focusing on the RNA-seq that we developed. So here is a overview of the flowchart of how RIMA runs. So there are actually multiple RNA-seq analysis tools and methods developed for so many years. And also many platforms such as the Galaxy, the Terra, and Cyverse, that's platform for providing the pipeline analysis for the samples. Most of those platforms, they were able to support small-scale data analysis at individual level and not able to conduct a really well cohort level downstream analysis. And we may also see like the problem focusing of the RNA-seq pipeline might be the trend of the future. So in here, we actually developed this immune-focused analysis pipeline, the of RIMA that aim to systemically processing the trials from the CMAC CIDC network that will be later used for cohort level analysis, and try to automatically solve the downstream analysis according to the PI's requirements from the very complex clinical meta sheet. So here's the flowchart. Data processing is a very regular process of RNA-seq. And then we have the 7 downstream immune-related modules. For example, the differential and gene set enrichment, the immune response prediction using the well-known gene signature markers, as well as looking at the cell components estimation of the cell type infiltration, and also looking at the TCR and the BCR repertoire from the RNA-seq data, as well as the fusions, the neoantigen part, as well as the we try to look for the microbiome data from the RNA-seq. So the RIMA will support the both individual and cohort level analysis. We also create a dynamic report for the user to visualize the result. So the pre-processing module is quite standard. We have we use STAR to do the read mapping, RCQC to check the quality of the data and the read alignment. We use SAMN to do the gene quantification, and we put many effort to working on the batch correction part, because from several of the CMAC trials, we did observe the the batch effects may be caused by the tissue type of the frozen tissue or the FFPE sample, or these samples can be sequencing at a different site of the CMAC center. This could be all the reasons that will cause the batch effect. So we use this PCA plot to visualize the data to see if there is any batches within the samples. Then we need to correct the samples before we put the expression matrix into several downstream analysis. And we also incorporated the features that is related with the data quality. For example, the unique mapping rate of the reads, and the total number of reads that is enough, the sequencing deep enough, as well as the ratio of the codon sequence mapping, as well, and the integrity of the sample. In the differential module, we conduct a differential expression analysis using the DSEQ2. And if we observed a batch effect, the factor will be added as a covariate in the DSEQ2 model for the differential gene expression analysis. After we identify the expression change, we are able to use the genes for the gene set enrichment and the single sample gene set enrichment. The other part we are incorporating is that we are actually looking at the expression profile of each sample using a hierarchical clustered heat map to look at the changes between the clinical group that we're interested. Here is an example of the changes we try to see, as well as the log-fold change in the responder and non-responder group. According to the GSEA, we did the annotation through the KEGG pathway database, the gene ontology, and we also incorporated the hallmark gene sets from the MCDB, which is a coherently expressed signature that is aggregating by the molecular signature database. This may also contains the gene signatures is immunotherapy related markers. TIDE is a gene signature prediction tool developed by the alumni Peng to calculate the gene signature scores describing the T cell dysfunction and the exclusion level according to the cytotoxic T lymphocyte level. So this TIDE is incorporating both the dysfunction and exclusion. And we actually observed this can be a very decent marker in predicting the patient response. As you show here, shows the highly correlation with the actual responders. And GeneSync in our lab developed this database source that collecting the public ICB data, ICB treated data, RNA-seq data from the public resources. And we use the area under curve to evaluate if this tide marker is a good prediction of the clinical response. We actually observed that tide works well on the melanoma cancer, but it did vary, depend on the cancer types. And here we highlighted the 3 trials that we collected with a decent AUC. So in our immune response module, we incorporated the tide and the MSI marker for the immune response prediction. In tide, we are not just predicting the dysfunction and exclusion. We are also looking at some common gene signature markers can be estimated from the tumor expression profile. For example, the cancer-associated fibroblast, the myeloid-derived suppressor cells, and the macrophage level, as well as the gene markers from the interferon gamma pathway and the individual gene signature markers. Using from the MSI prediction, there are many tools developed to predict MSI from either RNA or DNA. Previously, most of the tools will only support the DNA data from the tumor and normal sample. But the real case in CIDC is that for RNA-seq, we don't actually have the paired tumor and normal sequenced. So we actually applied this recently tool called MI-Sensor2 that can consistently detect the MSI level from both RNA and DNA and only requires the tumor-only data as an input. And so at the cohort level analysis, after recalling the MSI level and the tide level for each individual at the cohort level, we also did the similar group comparison of those well-known markers between the patient group that user can define. Here we show the example that within the comparison between responder and non-responder. The next is the immune infiltration module. That's currently there are 7 algorithms incorporated to infer the immune infiltration from the expression profile. Immune-deconf is a package that incorporating the CyberSort, timer, QuantiSeq, Excel, Epic, and MCP counter, the 6 tools. Timer2 is another tool developed by our lab that can estimate the immune infiltration level by looking at the closed cancer type from the TCGA. Because timer 1 requires you to give the cancer type, but timer 2 will estimate that for you. Actually, you can see different tools will estimate different cell type. That is because in their algorithm, they use different sets of curated gene signature immune markers using their own methods. So they may results from the 6 number of cell types and the results from the 6 tools there could be different. In RIMA, we not just estimate the infiltration level, but we also looking at consistency of these 6 tools using the common cell types that's been identified. Here we can see here that we use the cell types, the B cells, the CD4 T cell, CD8 dendritic cell, macrophage, Tregs, NK cell, and neutrophils. So by using this, we're able to evaluate the consistency of the cell types that's being identified through the 6 tools. That will give us a more high confidence on the result. On top of that, the cohort level analysis, we are also trying to look if the infiltration is significantly differences within each cell type between the patient group comparison. Here, the example shows that we did find a significance according to the Wilcoxon test, that in the activated mast cell, there is a significance in the immune infiltration. Immune repertoire could be another important factors that is associated with the clinical response. And previously, TCR-seq and BCR-seq is used to calculate the immune repertoire, but those data may not be widely accessible compared to RNA-seq. So Lee from our lab developed the Trust4 that is published recently that can effectively infer the immune repertoire from the RNA-seq data and the single-cell RNA-seq data. It's also been tested on the TCR and the BCR-seq as well. But compared to other tools that mainly developed using TCR and BCR-seq, Trust4 is able to identify higher context and more CDR3s from RNA-seq data. And if you're interested, you can read the Trust4 papers as well. Using the Trust4, we're able to identify the CDRs, and using these CDRs, there could be several metrics we could use to estimate from the clonal, if after we cluster the CDRs into several clones. For example, we are able to calculate the diversity and the clonality of BCR and TCR separately. We're also able to check like how many unique clonal types were identified in each sample. And for BCR, we are also identified the somatic hypermutation according to the alignment of the CDR3 to the reference database. Besides that, Trust4 also infer the isotypes of immunoglobulin. So in here, this plot, we plotted the ratio of the Ig composition that could represent the different maturation status of each samples here. At this level, we are also able to do the comparison to see the difference of the each Ig composition from the patient group. Another part of neoantigen module is also quite very important, that is associated with the HLA presentation and the neoantigens that could affect the treatment of the blockade. Actually, we are not tend to identify the somatic mutations from the RNA-seq data, but we are able to identify the HLA types using RNA-seq data, and the recent tools developed is called ARCUS HLA, and it's been highly it's developed specifically for RNA-seq, and it's highly correlated with the other tools. For example, the OptiType that has been widely used on the DNA, but the result using the RNA and using the result of ARCUS HLA on applied on RNA-seq is highly consistent with the result obtained by OptiType on DNA data. So in here, we use the ARCUS HLA to identify the HLA alleles. As I mentioned, we also have the whole exome pipeline that will give us the somatic mutation result. Incorporating the
expression. If you have whole exome, why do you need the ARCUS HLA then? If you have whole exome sequencing, can you just call the HLA from the whole exome?
Yes, yes, exactly. But as I mentioned, like whole exome data using the OptiType, which is also requires you to have the tumor normal paired input that will gives you the HLA result. The case in CIDC, we did find many samples only has the tumor sample. So in that case, if you have the adjacent RNA-seq data, we can use ARCUS HLA for that.
See. And again, I guess I asked you before, the problem of trying to get the somatic mutations from the RNA-seq, is this too noisy?
Yes, it's very noisy. If we want to decrease the noise, we have to have a deep sequenced RNA-seq data, but still, I don't think it's recommended to.
Yeah, just often you're going to only have RNA-seq and no whole exome sequencing.
Yes, yes. So at this point, within CIDC, we may tend to use the somatic mutation from the whole exome rather than calling it from RNA-seq. By the way, we don't have the tumor normal paired RNA-seq data, so we cannot correctly call the somatic mutation from it.
I see there's not enough just normal variation known now. I mean, there's a lot of normal sequencing done already. Shouldn't we be able to tell without the normal what the normal should be? Yes,
then that will be related to create a panel of normal mutations from the public normal set.
Yeah, but there's, must be a million normal sequencings done already. We must have a pretty good knowledge of what the normal variation looks like across people.
Yes, there will also need some efforts and validation to create a high confidence panel of normal that can correctly calling the good quality somatic mutation. Within CIDC, the whole exome team is actually working on that to try to use the normal data to create.
Yeah, because it seems now that we should have enough normal to know what the possible normals are.
Yeah, exactly. So we try to work on that using the normal samples that we already collected and the public ones, but it still needs some effort for it. Yeah, because actually in the West data for those tumor only samples, we need the panel of normal for it to call the somatic mutation. Yeah, and continuing from that, so if we have the somatic mutation either from the RNA-seq or DNA or tumor only mutation you called using the public normal SNPs, we can incorporate these two information into the tool called PVAC-seq to identify the potential new antigens. So what PVAC-seq do is that it calculates the binding affinity of the peptide fragments of those of the mutated peptide fragments and the wild type peptide fragments, incorporating the results from multiple tools. the net MHC-PAM, MHC flurry, and SMN align. But actually, the net MHC-PAM is the most commonly used, and it's been developing using latest computational techniques. So I think some others already actually studied net MHC-PAM may be already good enough for predicting the HLA binding affinity. So as I mentioned, the PVAC-seq can identify the binding affinity of the mutated peptide fragments as well as the Y type. Sorry, the Y type. So in here, when the common criteria people use to identify if this is a new antigen, that is we have to have the strong binding affinity in the mutated fragments, and also you didn't observe a binding affinity in the adjacent to Y type. So IC50 value is the criteria to determine if this binding is strong enough or not. So after this, we are actually able to find the novel and non-novel new antigens as well as the genes where the somatic mutation happens. Even though we're not able to, we could, but at this point we didn't call the somatic mutation from RNA-seq data, but we could call the gene fusions from RNA-seq data. So STAR fusion is the commonly used tool to call the fusions that's using the output directly from STAR. So we use this STAR fusion to call the gene fusions from the alignment results of STAR. From the trials that we have in CMAC, we did find there are still many homologs, or those are false positives called by STAR fusion, and the sequence similarity is quite high. So we actually used another tool called PyPrada to detect the homolog genes of the that shows the high sequence similarity. And so these genes will not be included in the downstream cohort level analysis. But rather than that, we did a annotation of the gene types according to the annotations we collected from OncoKB. OncoKB is another knowledge base that saving the oncogene information. So in this way, we are able to identify if this gene is a cancer driver gene or tumor suppressor gene or oncogenes. So we try to incorporate all the informations in the cohort level plots. So we actually did plot the expression level and the gene fusion in the responder and non-responder group, as well as using the different colors to represent if this what type of genes this is. Microbiome is also a hot field in the immune that many labs. Microbiome has been studied for a while, but incorporating microbiome with immunotherapy has also been started by labs. And actually, there are many papers where we identify some bacteria that is particularly associated with the response or the side effect of the ICB treatment. 16 people usually do the microbiome using the 16S or the metagenomics. Tools that particularly designed for calling the microbiomes like centrifuge and Kraken were used for NGS data to call the microbiomes. In here, we use centrifuge, which is more rapid, is an efficient classifier, and it works better for the RNA-seq data. So we use this centrifuge. centrifuge were able to classify the RNA secrets back to the pre-built bacteria and virus reference database, though in the real case is that we were not able to identify high abundance of those bacteria from the RNA-seq data, especially the RNA-seq data with low quality. But still, there could be hundreds of microbiomes identified by that.
Did you add it back? You have all the viruses now? You have a virus file as well? Because those are all back. You have the viruses in the database? Because you're more likely to find viruses, I think, than bacteria in most tumor types. Like it's gonna be hard to find much bacteria in breast cancer or melanoma, but you're likely to find viruses in some cancer types.
Yeah, that could be. In our reference, we didn't split that, but that could be good way for us to check if we are more focusing on the virus. But now we didn't actually look splitting the bacteria and the virus. But yeah, but that could be a good direction we go to look.
In the colon cancers, you might find, the GI cancers, you're going to find lots of bacteria, but in the, I think in the melanomas, you're not going to find very many bacteria.
Yeah, exactly. Also, yeah, so this could be virus, varies a lot. We know like people not usually looking for microbiome in the RNA-seq way, the shotgun or the 16S were widely used, but we still like incorporating this feature in the RIMA pipeline. So like hundreds, yeah, so usually there could be hundreds or thousands of microbiomes. And for the visualization purpose, we did use the inverse Simpson diversity score to actually only plotting the top 15 mostly enriched microbiomes in the samples. And later, we were also incorporating the diversity analysis of the microbiomes and also the phylogeny analysis. This could be the future directions we want to dig into microbiome, but we still need some solid data that some more solid data to show us that this actually functions really well in the RNA-seq. So one issue that when we are in the real analysis of the CMAC trials is that there could be very complex clinical sample and people want to do comparison from multiple different groups for saying the benefit versus no benefit, pre versus post, responder versus non-responder, or at different time points of the treatment, or the combination of the chemotherapy and the immunotherapy. So could be very complex comparisons people want to do, and the different number of samples included in the data. We try to find a better way to support that. So we use this group called, it can be any column that defined by the user to control what kind of samples you included in this comparison and what's the treatment and control we want to add. So in this way, we're able to conduct multiple cohort level analysis comparison using the RIMA. At the same, now we do each run for it. We do the individual run for individual comparison, but later we could incorporate the multiple group comparison running for the same time. The other thing that RIMA is incorporating this dynamic HTML report for the individual and cohort level analysis. We try to integrate as much as result on cohort level analysis here is an example. I'm going to demo example and show you using the data from this glioblastoma treated with the anti-PD-1. And we are going to show a example of six samples of 3 responders and 3 non-responders. Here, this is a simple HTML report. Only one integrated files, HTML files, will be generated. In here, we save the basic information of our pipeline and the tools of the versions that we use, as well as the meta sheet. You can play this sorted by the group that of your interest. In here, we show the comparison of the group of the responder and non-responders of the only pre-treated responders and non-responders. And this is the quality metrics that's incorporating the 4 metrics here. Now I'm actually sorting by the unique mapping rate. And if there is a sample with very low quality, it's very easy for you to see. And in the differential analysis, we plot the I'm also sorted by the significance. And you can also sort by the log4 change if you're only interested in the positive or negative log4 change. In this one, we only show the top 100, but actually when you run the pipeline, you were able to see the whole text result. And here we incorporated the volcano plot showing the most significant differentially positive and upregulated genes. In GSEA, we're actually able to identify many gene ontology or the pathways. In here, we only show we display the top 10 that is with the most significant enrichment score, as well as the single sample. As well as the single sample results according to the KEK pathway, you can see if this gene is highly enriched in this pathway or not, according to by the positive and negative values. Immune infiltration module, we show the exact immune infiltration level of all the six components here. And actually, we use this matrix to do the comparison analysis as well. Under immune repertoire, we do the comparison of the 3 responders versus non-responders according to cell type, different metrics. In here, in the BCR, one of the samples, we didn't have enough clones identified, so we're not able, actually not able to get the somatic hypermutation, so the result is not shown here. And the LG frequency that we did as I mentioned, that if you mouse over, you actually see the actual ratio of the different immunoglobulins you found in different samples. And here is what you see in the immune response. TIDE is a cohort level analysis, so we only incorporated the 6 samples we included in this comparison, and then we also gives you the comparison significance of the TIDE MSI exclusion dysfunction, the CD8, CD274 level of all the cancer-associated fibroblast and the MSDSCS level. This plot actually shows you the trends from your data and how it compares to the TCGA data. So once you we're looking at the glioblastoma in TCGA and do the similar calculation of those well-known markers and try to compare the patterns of it. In the neoantigen, we just use a table to show the exact HLA alleles of the sample. And in the fusion, here is the plot that we just displayed. We are also planning to incorporate the table that shows you the exact fusion genes and what is a genes. And here, the genes we marked here is actually the target genes that we used for the gene annotation. So for the genes, so you will only see one annotations here. Here another upgrade that we have, that we will allow the very flexible module level or individual and cohort level control of the RIMA pipeline. And we already tested the RIMA on the AWS, Google Cloud, Amazon Cloud, as well as the CIDC high performance computing, the Kraken. So we can successfully run the data from the 3 platforms. So thanks to the SnakeMake, the SnakeMake allow us to package the environment file that is needed for setting up RIMA. So we packaged the environments needed file, and then we just need one simple command, and you can install the RIMA environment within 10 to 20 minutes using the Mamba and using Conda and Mamba. I'm going to show, we have a tutorial with text and video tutorial in Gitbook. I can show you later. And here is the example configuration YAML file. That's for us to control what kind of modules you want to run, and it's an individual run or it's a cohort level run. You just need to set it to true if you want to do the comparison. Here is if you're interested, here is the link. You go to the liulab GitHub and go to RIMA, and it will show there is a detailed description about how you can run RIMA on instance, how you do the setup, environment setup, and as well as the detailed explanation of each module, how it works, how we calculate the for example, the coloniality and the diversity after we get the result from immune repertoire, and how we did the group comparison using the immune infiltration results, and the response, the HLA fusion, and the microbiome. So we also give you the instruction of the steps if you want to create your own reference. For example, if you want to run RIMA on mouse, what kind of reference file you need to prepare, and then running RIMA on it. I know some in our lab you will also use RIMA to run the mouse models as well. So in other words, RIMA can be customized to other species.
So do we have the mouse reference files done already?
We have one version that we use on Kraken. We could probably integrate that in the next version of RIMA.
So I think both mouse and rat would be very useful for us. So, you know, Nelly's lab and
Yes.
doing.
Yeah, that's what I wanted to say, that the rat is very complicated because it's so under edited, but this is something we really want to do.
So both mouse and rat would be very useful to have the reference files for.
Yeah, the mouse is easier than rat. Rat, even the sequence isn't, but we have whole genome sequence. So we have all the data, but we just need the pipeline to annotate it. Yeah.
Yeah. I think we have the mouse, but we don't have the rat yet. But actually, it's very easy to build the index. But I believe most of the tools they were only developed for humans. So if we want to apply the tools to mouse, we may need some gene ID conversion.
Yeah, I think that's for mouse to be really straightforward. As sure, as Nellie said, I think for RAT, it's going to take more work, but I think if we need to find someone either in your group or in the CFC who's willing to take on this rat and mouse conversion, because I think for the models, it's extremely useful to have both of those.
Yeah, I have a new postdoc starting in October. He can do some computation also, but we certainly would need your help. But we have lots of, we have the whole genome sequence data, we have RNA-seq data, we're gonna generate more data. So I think we can
There's more about these tables and reference files for RAT.
Yeah, for RAT, even the genome sequence was somewhat complicated because there are 3 different ones and it was not a perfect match.
Yeah,
which was really irritating.
Yeah, in that case, might need more effort to do the annotation or something.
Yeah, but maybe we can, again, I think mouse is going to, mouse, all of your members of your consortium are going to want to have mouse for sure. People like Kai and these people, they do a lot of mouse work. So I think mouse has got to be something you definitely, offer, that you have packaged in the container.
Yeah, we're doing a lot more mouse also. So immune competent mouse.
Yeah.
But the rat would be also useful. I may even, I don't know if Jos Jonkers has been thinking about that, because I just feel like we need a coordinated effort to, even the TCR sequence is not established in the rat. So
rat's definitely going to be much more of a challenge, but mouse is definitely.
The mouse should be more straightforward.
Yeah, we have a pre-built reference of mouse, and later we can integrate that in our GitHub as well.
Yeah, I think so. I mean, again, we're happy to have manpower to help with these. I think because I think these are definitely worth the effort.
Oh, that would be great, because I know CITC is have people shortage.
Yeah.
Okay. We can help. We have bioinformatician here, and we are happy to help and provide support for the assemblies.
Yeah, I think if the center, Gentile, if the center can offer help for the mouse and the rat, that would be very helpful.
Yeah, we are happy to help. Yeah.
Oh, that would be great. Probably is Tommy here? We can get in touch and start working on that.
Yeah, I think we're doing mouse experiments all the time, and people at the institute are doing mouse experiments all the time. So again, rat is a more long-term aspirational thing, but mouse is an immediate need.
Yeah, mouse is immediate need, and but the rat we just need to solve it. Yeah.
Okay, now I'll continue. Just a brief summary of how the RIMA. So RIMA is a quite comprehensive, as you can see, and it's more like an integrative, immune-focused pipeline. And the individual level analysis of RNA-seq is quite easy, but what we put many effort is to make sure the cohort level is meaningful, correct, and is flexible to support. It's still hard to replace a bioinformatician, but we try to make our pipeline more customizable. And the RIMA is mainly developed using the SNCMake within R and bioconductor, SNCMake with Python, and the bioconductor using R, and also the multi-QC dynamic report system. So it's compatible through cloud and high performance computing cluster. So thanks for the feedbacks from others. We did has many future development and features that is on the list to add to RIMA in the future. Many thanks to Shirley for providing good suggestions regarding the RIMA and the features that are important to add for the immune-related analysis. Also, Jan, Lin, Ashna, and Jing that first developed and develop the pipeline, and also makes the pipeline products more user-friendly and more impact. And also Li, Jing Xin for their help on the tools that exactly how to run, and Ashoro actually helped us to prepare the mouse at the beginning. Also, some other members from CIDC and the lab that gives us good suggestions. Yeah, thanks.
Really great work, guys. It's an amazing pipeline. I'm sure that you will get a lot of use. People will start installing it, using it, I'm sure. So Quintal, are we have it installed in the CFC server, or is that?
We haven't installed it right now, but it's on to-do list. As long as we, feel it's, ready to do the deploy version, because last time we have some suggestions. We want to do multiple comparisons at one time.
Yeah.
Some small changes. As long as we have the go signal, we can install it quickly.
Yeah, we had some input a month or two ago, and I saw a lot of that in the report. It's a beautiful job putting on these great new functionalities, and I love seeing it presented in that great report. So I think it's looking wonderful, ready to move ahead.
Yeah, and I do think the center can help with making sure the mouse is available. And I do think, as a long-term goal, adding RAT is going to be useful as well.
Yeah, we can make the CFC as our kind of beta tester, and I'm sure in the process we need to continue to improve and update features and also make it more foolproof, and then we can maybe deploy it with MBCF or like within a Farber.
Yes. Great. Tremendous job.
Yeah.
So, again, for me, seeing this sort of professional pipeline is what is often lacking. You see so many people publish bioinformatics stuff that no one can use, that never gets deployed because it's all hacked and not really done in a careful way. So this is extremely satisfying to see.
Yeah, we try to make it more useful. That's why we spend many effort on to support the analysis.
Yeah. And it doesn't put the bioinformaticians out of business. It just makes them not have to do the repetitive work. The key is that all the repetitive work can be done if you make a pipeline. It's that leaves the more interesting work for the bioinformatics people to work on.
That's true.
The repetitive work should be done by the computer, not by the people.
Yeah, right. Okay, great. Yeah, thank you guys.
Thanks, Tommy.
Thanks.
Thank you.
Thank you.
Bye.